Hello dear students. Today we will discuss about the spectral analysis of heart rate variability. In last lecture we have studied about the different time domain measures for heart rate variability, and in this lecture we will discuss mainly the frequency domain measures for heart rate variability. Frequency domain method of analysis is one of the key factors for heart rate variability analysis. Various spectral methods for the analysis of the tachogram have been applied since the late 1960s. The most common power spectral estimation used in HRV analysis is the power spectral density, or we can say PSD analysis. PSD estimation provides the basic information of how the power of the signal, that is, its variance, distributes as a function of frequency. Frequency domain measures extract frequency information by using fast Fourier transformation, that is FFT analysis, or it can also use autoregressive modeling. There are various methods to calculate power spectral density. So there can be non-parametric methods and the parametric methods. The advantages of non-parametric methods are the simplicity of the algorithm, that is mainly we are using fast Fourier transform, and also it has the high processing speed. And the advantages of parametric methods are they produce smoother spectral components which can be distinguished independently of pre-selected frequency bands. Easy post-processing of the spectrum with an automatic calculation of low and high frequency power components and easy identification of the central frequency of each component. Also, it produces an accurate estimation of power spectral density E1 on a small number of samples on which the signal is supposed to maintain stationarity. The basic disadvantage of parametric methods is the need to verify the suitability of the chosen model and its complexity, that is the order of the model. Now we'll discuss the spectral components. Short-term recordings. Three main spectral components are distinguished in a spectrum, which are calculated from short-term recordings of around 2 to 5 minutes. They are the very low frequency, that is VLF components, low frequency, that is LF components, and high frequency, that is HF components. Measurement of VLF, LF and HF power components is usually made in absolute values of power, but the low frequency and high frequency components may also be measured in normalized units, which represent the relative value of each power component in proportion to the total power minus the VLF component. We have some absolute measures, that is total power, which is measured in 0 to 0.4 Hz. Then we have ultra low frequency band, that is ULF, which is less than 0.003 Hz. Then we have very low frequency components or very low frequency band, which lies between 0.003 to 0.04 Hz. Then we have low frequency band, which lies between 0.04 to 0.15 Hz. And then we have high frequency, band, that is 0.15 to zero point 0.4 bands. So these are the different absolute measures. Then we have some relative measures. The representation of low frequency and high frequency in normalized units emphasizes the controlled and balanced behavior of the two branches of the autonomic nervous system. The normalization minimizes the effect of changes of total power on low frequency and high frequency components, and they are given as low frequency, that is LF norm, that is LF divided by TP minus VLF, that is total power minus very low frequency components, and this will be calculated in normalized units. Then we have HF norm, that is HF divided by TP minus VLF, and also it is measured in normalized units. And another parameter is ratio of low to high frequency power, that is LF by HF. Then we have long-term recordings. The spectral analysis may also be used to analyze the sequence of NN intervals in the entire 24-hour period. It includes an ultra high frequency component, and it is represented as ULF. The slope of the 24-hour spectrum can also be assessed on a log-log scale by linear filtering the spectral values. This table lists various frequency domain measures. As you can see, we have 5-minute total power, that is measured in milliseconds square, VLF, LF, LF norm, HF, HF norm, and LF by HF ratio. So these are the short-term recordings we have already seen. Then we have long-term recordings, that is analysis of our entire 24 hours. In this again we can have total power, we can have power in the ultra frequency range, we can have power in the very low frequency range, we can have power in the low frequency range, high frequency range, and also we have another parameter that you can see, this alpha, that is slope of the linear interpolation of the spectrum in a log-log scale, and that is approximately in the frequency range, or which is less than or equal to 0.04 hertz. So let's discuss, or let's see about the interpretation of these frequency domain parameters. So what is total power? Total power is a short-term estimate of the total power of power spectral density in the range of frequencies between 0 and zero point 0.4 Hz. This measure reflects overall autonomic activity, where sympathetic activity is a primary contributor. Then we have power in very low frequency band, and it is a band of power spectrum range between 0.003 to zero point 0.04 Hz. It indicates overall activity of various slow mechanism of sympathetic function. In short-term analysis, VLF does not provide much meaning, since this band often reflects meaningless noise signals. VLF band provides an additional indicator of sympathetic function. Then we have power in the low frequency band. Basically, it is a band of power spectrum that is ranging between 0.04 to 0.15 Hz. We have already discussed about that. And this measure reflects both sympathetic and parasympathetic activity. Generally, it is a strong indicator of sympathetic activity. So parasympathetic influence is represented by low frequency band when respiration rate is lower than 7 breaths per minute or during taking a deep breath. Thus, when subject is in the state of relaxation with a slow and even breathing, the low frequency band power values can be very high, indicating increased parasympathetic activity rather than increase of sympathetic regulation. Then we have power in a high frequency band, so that range between 0.15 to zero point 0.4 Hz. This measure reflects parasympathetic, that is the vagal activity. It is also known as the respiratory band as it corresponds to NN variations that are caused by respiration. Heart rate is increased during inhalation and drop during exhalation. Generally, increase in high frequency power accompanies the increase in heart rate variability. Then we have LF by HF ratio. This is a ratio between the power of the low frequency and high frequency bands. This measure indicates overall balance between the sympathetic and parasympathetic systems. Higher values reflect domination of the sympathetic system, while lower ones reflect domination of the parasympathetic system. This ratio can be used to quantify the overall balance between the sympathetic and parasympathetic systems. Then we have normalized low frequency, and that is mainly we are measuring in percentage, and it is represented as LF norm. It is a ratio between the absolute value of the low frequency and difference between total power and very low frequency. This measure minimizes an effect of changes in very low frequency power and emphasizes changes in sympathetic regulation. Then we have normalized high frequency. It is the ratio between absolute value of the high frequency and difference between total power and very low frequency. This measure minimizes an effect of changes in very low frequency power and emphasizes changes in parasympathetic regulation. Now let's see what are the different correlation and differences between the time and frequency domain measures. When analyzing stationary short-term recordings, more focus will be on the physiological interpretation of the frequency domain measures derived from the same recordings. However, many time and frequency domain variables measured over the 24-hour period are strongly correlated with each other. This table approximate correspondence of it basically shows the approximate correspondence of time domain and frequency domain methods applied to 24-hour ECG recordings. As you can see, we have time domain variable SDNN, that is standard deviation of NN intervals, which is approximate correlate to total power. We have HRV triangular index, that is also correlated with total power. TINN, that is also correlated with total power. Then we have SDANN, which is correlated to ULF. Then we have SDNN index, which is correlated with mean of 5 minutes total power. And we have other parameters like RM, SST, SD, NN50 count, PNN50, differential index, logarithmic index. All these parameters are correlated with power in high frequency band. So these are the various parameters which we can see they are correlated in time domain and frequency domain systems. Now let's discuss about another topic, that is rhythm pattern analysis. The time domain and spectral methods have limitations that are imposed by the irregularity of the RR series, as you can see in this figure. So there are so much of irregularity in these waves. This figure shows four synthesized time series with identical means. All these have same means, standard deviation and ranges. You can see series C and D, they also have identical autocorrelation functions and therefore they can have identical power spectra. So in these cases, as we can see, different profiles are analyzed. They can give identical results, but actually they are not identical. Their pattern is not identical. So trends of decreasing or increasing cycle length are in reality not symmetrical, as heart rate accelerations are usually followed by a faster decrease. In spectral results, this tends to reduce the peak at the fundamental frequency and to enlarge its basis. This leads to the idea of measuring blocks of RR intervals determined by properties of rhythm. Approaches derived from time domain and the frequency domain measures have been proposed in order to reduce these difficulties. The interval spectrum is well adapted to link RR intervals to variables defined on a beat-to-beat basis, for example, the blood pressure. The spectrum of counts is preferred if RR intervals are related to a continuous signal, for example, respiration, or to the occurrence of special event, that is, for example, arrhythmia. The peak valley procedures are based either on the detection of the summit and the nadir of oscillations or on the detection of trends of heart rate. The detection may be limited to short-term changes, but it can be extended to longer variations. Second and third order peaks and troughs, or stepwise increase of a sequence of consecutive increasing or decreasing cycles surrounded by opposite trends. The various oscillations can be characterized on the basis of the heart rate accelerating or slowing, the wavelength and or the amplitude. In a majority of short to mid-term recordings, the results are correlated with frequency components of heart rate variability. The correlations, however, tends to diminish as the wavelength of the oscillations and the recording duration increase. Complex demodulation uses the techniques of interpolation and detrending and provides the time resolution necessary to detect short-term heart rate changes as well as to describe the amplitude and phase of particular frequency components as the function of time. Now we'll start another topic, that is interaction with other physiological signals. The analyzed ECG signal should satisfy several requirements in order to obtain a reliable spectral estimation. In order to attribute individual spectral components to well-defined physiological mechanisms, such mechanisms modulating the heart rate should not change during the recording. The sampling rate has to be properly chosen. A low sampling rate may produce a jitter in the estimation of the R wave. Further, the baseline and the trend removal may affect the lower components in the spectrum. The choice of QRS fiducial point is critical. It is necessary to use a well-tested algorithm, that is, derivative plus threshold, template matching, correlation method, etc., in order to locate a stable and noise-independent reference point. We have seen various methods for detection of QRS complexes, so we can use the appropriate method. A fiducial point localized far within the QRS complex may also be influenced by varying ventricular conduction disturbances. Ectopic beats, rhythmic events, missing data, and noise effects may alter the estimation of the power spectral density of heart rate variability. Therefore, the proper interpolation or preceding or successive beats on the heart rate variability signals or on its autocorrelation function may reduce this error. Preferentially, short-term recordings which are free of ectopy, missing data, and noise should be used. We'll see what are the autonomic influences of heart rate. Although cardiac automaticity is intrinsic to various pacemaker tissues, heart rate and rhythm are largely under the control of the autonomic nervous system. The parasympathetic influence on heart rate is mediated via release of acetylcholine by the vagus nerve. The sympathetic influence on heart rate is mediated by release of epinephrine and norepinephrine. We have various components of heart rate variability. The RR interval variations present during resting conditions represent a fine-tuning of the beat-to-beat control mechanisms. Vagal efferent stimulation leads to reflex excitation of vagal efferent activity and inhibition of sympathetic efferent activity. The opposite reflex effects are mediated by the stimulation of sympathetic efferent activity. Efferent vagal activity also appears to be under tonic restraint by cardiac efferent sympathetic activity. Efferent sympathetic and vagal activities directed to the sinus node are characterized by discharge largely synchronous with each cardiac cycle, which can be modulated by central, for example, vasomotor and respiratory centers, and peripheral, for example, oscillation in arterial pressure and respiratory movements oscillators. These oscillators generate rhythmic fluctuations in efferent neural discharge, which manifest as short and long-term oscillation in the heart period. Analysis of these rhythms may permit inferences on the state and function of the central oscillators, the sympathetic and vagal efferent activity, humoral factors, and the sinus node. So spectral analysis of 24-hour recording shows that in normal subjects, power in low frequency band and high frequency band expressed in normalized units exhibit a circadian pattern and reciprocal fluctuations with higher values of LF in the daytime and high frequency band at night. These patterns become undetectable when a single spectrum of the entire 24-hour period is used or when spectra of subsequent shorter segments are averaged. In long-term recordings, the HF and LF components account for approximately 5% of total power, although the ULF and VLF components account for the remaining 95% of total power. Their physiological correlates are still unknown. LF and HF can increase under different conditions. An increased LF, again that is expressed in normalized units, is observed during 90, tilt, standing, mental stress and moderate exercise in healthy subjects and during moderate hypotension, physical activity and occlusion of a coronary artery. Conversely, an increase in high frequency band is induced by controlled respiration, cold stimulation of the face and rotational stimuli. It is important to note that HRV measures fluctuations in autonomic inputs to the heart rather than the mean level of autonomic inputs. Thus, both autonomic withdrawal and a saturatingly high level of sympathetic input leads to diminished HRV. So these were the various, we can say, physiological signals or various factors that can interact with the heart rate variability measures. So this was all in this lecture, and for this we can refer to the paper by Merrick Malik, J. Thomas Picker et al. That is Heart Rate Variability, Standards of Measurement, Physiological Interpretation and Clinical Use. That is by in European Heart Journal, volume 17, issue 3, and it was published very old paper, that is 1996. So this was all in this lecture. Thank you so much.